And you stayed exactly on time, so I didn't have the opportunity to use my gavel once again, but hey, there's still hope. Emil is an infectious disease physician with the Army, where he founded and directs some really groundbreaking work on antimicrobial resistance surveillance and research, MRSN armor, at Walter Reed. His topic is antimicrobial resistance and the use of big data by the Department of Defense, highlighting challenges for large health systems. Emil.
Thank you very much. I'm very honored and extremely honored and very excited to have the opportunity to address the forum. The Infectious Disease Society of America, the CDC, the European CDC, the World Health Organization, and many other societies consider the escalating crisis of antimicrobial resistance to be one of the greatest threats to global public health and our ability to deliver safe, effective patient care. And especially for the viewing members and the public on the web link, if you take nothing else from my presentation away, this is my soapbox, is that we are running out of antibiotics faster than we can develop them. And sometimes, the organisms are developing resistance faster than we can mitigate that. And what we sometimes take for granted is that the miracle of antibiotics makes most of all medicine possible. The elective surgery, hip replacements, and cancer chemotherapy. So what I'm going to talk about today is how the DoD attempts to use big data to fight antimicrobial resistance. And I realize I have three challenges. The first challenge is I'm the last thing between you and lunch. My second challenge is I'm going to be compressing 10 years of moderate or big data into 10 minutes or less. And I have to prevent Scott from gaveling me. And I forgot what my final challenge was. So the final challenge is, why should we care about what the heck the DoD is doing? As taxpayers, I would argue that you're our most important customers and stakeholders of the DoD. And in the spirit of transparency, we like to take these opportunities to remind you, the public, nonprofit organizations, and other government agencies that what we develop in our products are available to you. We also like to keep you updated as to where your tax dollars are going. Secondly, many of our products, although most of them are knowledge products, many of them are transferable. Our approaches, some of the tools, the assays we develop are readily transferable to the civilian sector. And finally, for this venue here where we're focusing on challenges, I'm going to say that if the DoD can do it, anybody should be able to do it. And what I mean by that is, one of the biggest challenges that we face with big data is access to the internet and moving that data around. And a highly secure .mil network has additional challenges that need to be overcome. But don't tune out. My argument is that large healthcare organizations, geographically dispersed healthcare organizations, are likely to face some of these very challenges. And in the end, I'll focus on what I believe are the most important challenges, and I believe that they're the most important challenges because they're not going to be solved by technological advances, and they're the ones more likely large healthcare organizations are likely to face. Indeed, you see that we're facing those challenges already in the form of cyber attacks, ransomware. So, although many civilian facilities have open .orgs or are less restricted environments, my prediction is that increasingly healthcare organizations will come under tighter restrictions and tighter controls, which will limit your ability to move, analyze, and process big data. So I'm briefly going to talk about the size of the data challenge in the DoD, do some examples focusing on the two main types of bacteria, gram-positives and gram-negative, and wrap up with some of the challenges that we faced using some of these examples. The size of our data challenge, there's about 9.2 million healthcare beneficiaries in the Department of Defense that are eligible to receive care each year. Each one of those can generate 1 to 100 cultures, and each culture can generate 1 to 20 results, amounting to about 9 million to 18 billion results per year. Now granted, 2/3 will stay healthy, not seek care, or not have an infection-related condition. However, we have these horrific injuries now sadly continuing to occur, where these patients are in the hospital over a year and can easily generate over 100 cultures each. So you chop that by a third and you have 3 million to 6 billion results. But here's where the big data challenge comes in, linking their whole genome sequences of all those culture results. That creates a tremendous amount of data. In the military, dogs are people too, so we espouse and we're highly aligned with the One Health, One World concept. So adding to our surveillance and our outbreak support, as military working dogs play crucial roles in explosives and drug detection or in healing, we support surveillance and outbreak investigation with that. This is a busy slide, but how we create and analyze big data in the DoD is summarized 3 easy ways. And this is about policy. This form has policy in the title. So the first thing we do is we leverage public health and infection control surveillance mandates. All Army hospitals are mandated to participate in the surveillance, and by this time next year, all DoD hospitals will be in all services. The second thing we do is we create specialized data marts or data warehouses that contain structured and unstructured data that can be queried and mined in real time to provide alerts and other things that I will demonstrate later. And then finally, in combining what's been referred to as the next revolution in infectious disease practice, whole genome sequencing, you have to establish a high throughput genome sequencing pipeline. How does it all fit together? This depicts the flow of isolates, the flow of data, and the resulting flow of actionable information in the DoD healthcare system. And the beginning and the end all is the hospital up there, and when the hospital identifies a target pathogen or thinks that they have an outbreak, alerts are sent out, they send the isolate to the central lab, but also ongoing. This is a very rapid, interactive, and reiterative process. You can see there. Notifications and alerts are sent out to policymakers. Various types of report, including personally identified information, are immediately fed back to the hospital. And so if we get into a little more, and we travel to the central lab where many of these isolates are characterized and archived, another way that we increase the size of our data flow, but another challenge that we'll talk about is addressed by this slide. The challenge is that, unlike research or genomics institutes where you most likely have highly motivated, stable staff, in clinical laboratories, clinical hospital labs, especially in the military, there's a high turnover rate. And library preparations for sequencing are very demanding. And sometimes by the time you train up an individual, that individual is rotated out. So one of the ways we cope with that, and the other way that we're able to process 300 to 500 bacterial sequencing specimens a month, is through the use of liquid handling robots. So at the central lab, we have 2 high-capacity liquid handling robots, 4 Illumina sequencing platforms, and 1 Pacific Biosciences platform. The structure of what we call the ARMOR database, Antimicrobial Resistance Monitoring and Research Program, is depicted here. What's key about this is that it links clinical demographic information to include personally identifiable information, but it's secured in its firewall. It links it to freezer location to manage the isolate repository, but it also links it to the results from the antibiogram, so the characteristics of the pathogen, but also to the whole genome sequencing. Using this high-capacity sequencing pipeline that we developed, it contains commercial, off-the-shelf software, open-source software, and some in-house developed software. What it boils down to is that whole genome sequencing, we can replace many of the conventional genetic fingerprinting tests, such as PFGE, MLST. We can, without the need for doing thousands of different PCR reactions, we can get the gene content of all the resistance genes, all the known resistance genes and all the known virulent genes from a single sequencing run. It identifies quickly any mobile genetic elements that are very important for the transmission of these resistance mechanisms, and it gives you not 1, but 4 even more different ways of determining relatedness or genetic fingerprinting. You heard this morning that one of the V's of the data is velocity. Using this pipeline, we consistently now deliver a 48-hour turnaround time anywhere in the world, so that when our lab is contacted for outbreak support, after we get the isolate, 48 hours later, they get a SNP-based, down to the highest resolution outbreak report. So, big data versus gram negatives. Our question here was, at the level of an entire healthcare system, at the time we did this, we didn't think that this was looked at an entire healthcare system. And what we wanted to look at was relationships between antibiotic use and a very important type of bacteria that you see in the news sometimes. It's the notorious one where the fatal outbreak at the NIH. Same type of bacteria associated with the deaths from endoscopy. So we looked at that, called CRE. Our data set included 75 million person years of surveillance and up to 2 million cultures from 266 fixed facility hospitals. We looked at it overall, and then we stratified it regionally, by facility, and by drug. And depending on how you slice and dice it, you get a bunch of different conclusions. But you might think that, so for example, when you're considering a use-resistance relationship, you would say, okay, probably the more penicillin I use, the more penicillin resistance I'm going to find. So when we looked at carbapenem use, it was not necessarily correlated with carbapenem resistance. But one thing that was interesting, if you looked at it by quinolones, fluoroquinolones, and you looked at inpatient use, we didn't look at it. This is what the big data analysis fell out. What fell out that was statistically significant was that inpatient quinolone use at the two major referring facilities, one in the Northeast and one in Texas, were correlated with carbapenem resistance, not fluoroquinolone resistance, but carbapenem resistance in one of the most common pathogens in humans, E. coli, the cause of urinary tract infection. So the data period was about 2005 to 2012. There was a significant increase and then a significant decrease, and that was concurrent with full maturation of this collection data monitoring program. Whether they were associated or related, you can't tell, can't be determined. You focus down on the worst of the worst, if you will. So that slide, the two previous slides, were just focusing on resistant phenotypes. This slide depicts the trend from 2009 to 2014 of the confirmed ones that are genotypically confirmed to produce an enzyme that degrades our strongest antibiotics. And there's a spike followed by a decrease. Another type of bacteria that is a huge problem for the military is Acinetobacter. And we wanted to look at what was the differential. Our question here was, what's the differential burden between the carbapenem-resistant Enterobacteriaceae, in other words, the national health species bacteria, or the endoscope, what was the difference between that and Acinetobacter? Because before the conflicts in Iraq and Afghanistan, the military was relatively unknown, Acinetobacter in the military. It became so bad that it was considered almost to close Walter Reed Hospital down at the height of the outbreak. But that was I should say, in quotes, that was just multi-drug resistant Acinetobacter. We took it up a notch and looked at what about carbapenem-resistant Acinetobacter, because carbapenems are currently the broadest, strongest spectrum of antibiotics that we have. The data set included, we were able to capture data from about 15 million cultures to identify 360,000 potentially carbapenem-resistant strains, and these were the statistically significant relative risks. If you had a bacteria that was isolated overseas or from the bloodstream, you were a lot more likely to be carbapenem-resistant. Although Enterobacteriaceae were more frequently isolated, compared to those, the Acinetobacter or the Pseudomonas were 73 and 210-fold more likely to be resistant to carbapenems. Whereas the action in that data, you said, well, one action is the healthcare providers are now alerted to the fact that if their preliminary report, if they're overseas and they get a preliminary report from the lab that says we have a gram variable non-lactose fermenting gram negative, you should consider this elevated risk of being carbapenem resistant and adjust your empiric therapy accordingly. Another example of how that's actionable is you can intensify or increase monitoring or surveillance effort for those areas or for those anatomic sites. Some rates decreased, some rates increased, but not so fast. So this is a perfect segue into a special challenge that we encounter when we're trending antimicrobial resistance over time. That is the definition of of what's susceptible and what's resistant to a certain drug. That's called a susceptibility breakpoint. Currently, at least 5 different organizations issue compendiums of susceptibility breakpoints. The FDA has one, UCAST has one, USCAST one, and they're not all the same. So what your conclusion can sometimes be affected by is whose breakpoints you're using. Additionally, these breakpoints are updated at various times, and they're never updated at the same time. So so you might be seeing a downtrend in resistance, and that might be purely a result of the definition of resistance, because the definition was made more stringent. The minimum inhibitory concentration, for example, dropped from 8 to 4 to 2. Deduplication is a problem, and adjudication. So a lot of these isolates, and especially at our central lab, are repeated on different platforms to replicate their original platform that they were tested on, and just other hospitals. The patient went from hospital A to hospital B, and it just so happens that they have retested the specimen. Oftentimes, especially for the most resistant ones, you don't always get the same results. So how do you adjudicate those things? All special challenges. Another example of how we leverage these data marts is looking at targets for better stewardship. And one of the most common reasons that people visit their primary care physicians is for acute respiratory infections. And acute respiratory infections are, in turn, one of the most common reasons or misuse of antibiotics. And we looked at the data set, the size that you can see there, and you can see who's prescribing more, what classes, and target your education and your intervention accordingly. Big data, not necessarily better data, as we see here, or this goes to the other V we heard about, veracity. So gram-positive Staph aureus, another very important healthcare-associated pathogen. Our goal here was to try to develop an approach or a toolbox for trending a particular type of Staph aureus. This type of Staph aureus eludes detection by the automated susceptibility machines. It doesn't elude detection, but what the problem is, it's susceptible. So there's vancomycin-resistant Staph aureus are very rare. Vancomycin-susceptible Staph aureus are quite common. But in between there is a category where they're technically susceptible, but we call them clinically problematic. And if they have an MIC right around 1.5 to 2, that has been shown to have worse outcomes. The problem is, to get at that level, you need to do very laborious, highly specialized manual broth dilution tests, which is not feasible for large healthcare organizations or for hundreds of thousands of isolates. So we wanted to develop a semi-automated tool to see if we can do that. We used some of the approaches that the previous speaker mentioned, seasonal trend decomposition and autoregressive modeling. And the data set was about 230 million patient encounters, about 6.5 bacterial cultures, and from that we identified and deduplicated about 81,000 unique Staph isolates. Now in the DoD, they're tested on the 3 most widely used platforms. One platform is not used across the entire healthcare system, but that's similar to what many large geographically dispersed healthcare organizations use too. So if we could develop this approach, it could be translatable to them. So about 58% are tested on that platform, you can see there, 14 and 20% on the microscan. What did we find? Well, we found that we were able to do this and predict this tool. In addition to what we wanted to do was we wanted to have the tool to not only determine what the incidence or what the point prevalence of those problematic Staph aureus were, but also to predict their trend in the future and to do the same for drug use. So we were about ready to issue a report on this and say, well, you know, it could be concerning because it looks, based on like this ARIMA, that the trend is increasing. And a question this morning was, how do we display uncertainty? So the shaded lines and the darker shaded lines are the 95 and 99% confidence intervals of the prediction where that trend is going to go. Now, people that our bioinformaticists, the great group of team of data analytics that we have, looked at that and said, Lesho, I don't trust it. I don't like the data because it could be spurious. But my argument to them was, okay, so if that's being driven by a quirk of one platform, that quirk will be consistent across the study period and it'll cancel each other out. And furthermore, the platform that we were worried about, only 20% of the isolates were tested on that. So we said, nah, it's not going to matter. But indeed, I was wrong, it did. The actual trends are flat. So one of the things we do is for that platform, for Staph aureus, you can't use it for trending. And this is a starker example of what happens. So about 54% of the isolates would be technically problematic by one platform, but at real value and consistent among the other two, it's actually only 2%. And you see how the incidence or the prevalence dropped. That's the same facility that switched from platform A to one of the other platforms. The trends here are the real trends. Everything is a monotonic downward trend. So one of the challenges here we have, what was also alluded to, was the lack of variety. So basically, most of the drugs were decreasing. The trends in the Staph aureus were decreasing. The trends in the subset of Staph aureus, and there were not even any major spikes. So we lacked the variability to more thoroughly study association and causation. Maps, we heard everybody likes maps, so we can plug the data into a map and show that the larger the size of the circle, the more common the problematic type of Staph aureus is at the military hospital. And if you switched out Staph aureus and if you switched in CRE or carbapenem resistance, that would be completely different. From the northwest would go away almost to zero, and the mid-Atlantic and the northeast would be a huge circle. In the interest of time, so for large healthcare organizations, I summarize the challenges, the most important challenges, in 4 parts: generating, sharing and storing, and analyzing. And just so what we've also, Moore's Law, you've all heard of Moore's Law, where the computing processing speed and stuff is rapidly advancing. Well, this is a big factor in next generation sequencing. The advances occur in next generation sequencing in their kits, in their library preparations, in their platform, even faster than computers. But for public health and government-funded laboratories, or laboratories that are not funded with R&D funds, but a special type of money that's called operation and maintenance, the acquisition procedures are very long and very lengthy. And some of the problems that we've encountered by the time we put in the request and the award, and I'm gavelled. So let me, you can read that in your handout. And generating lengthy, burdensome processes, storing, we generate a terabyte of data a month, and every time we ask our commanding agency, can we have more space, we have to do a business case analysis. That's not sustainable, and analyzing and sharing, and I'm sure others will expound on that. This is the wonderful team, and these are my acknowledgements, and thank you very much.